Welcome to Unit 11, Microbiology. This is one of the more fascinating subjects for students, learning all about disease-causing organisms, the pathology behind it, the treatment, and especially the identification of these little critters. Microbiology is the branch of medical science concerned with the prevention, diagnosis, and treatment of infectious diseases. Microbiologists study the characteristics of pathogens of the 4 types of microorganisms that cause disease: bacteria, fungi, parasites, and viruses. Most small medical micro departments will focus on bacteria, while others that are very large will also include fungi, parasites, and viruses. Work performed in the department include determining the suitability of samples submitted. Our results are only as good as the samples that are submitted. And you have to follow the rules. There's certain steps that must be taken in order to get a quality sample. They will culture, and this is done by plating samples obtained from patients onto appropriate growth media. And this is called agar, and it looks like jello, but it has nutrients for the different kinds of bugs. The media is observed over several days for the growth of potentially pathogenic organisms. We will then perform testing to determine the identity of the organism, what is your name, and then test the organism against drugs known to kill the organism to determine which one is most effective, because as you've heard in the news, many bacteria are becoming drug resistant. So we run the bug against a panel of drugs to see which drug is the most effective at killing the pathogen. Anything that comes out of your body can be cultured in the microbiology department. The most frequently submitted samples are blood, urine, throat, urogenital samples. Usually we're looking for sexually transmitted diseases, stool, wound, and sputum. And sputum is the sample that I had the most difficulty working with because it's not saliva. You tell the patient to breathe very deeply and cough up what's very low in their lungs. And yeah, it just gagged me every time I had to plate those. There are many other samples that may be submitted. The types of samples cultured will be dependent upon the type of facility that you work at. Normal flora is defined as bacteria found in or on specific areas of our bodies without causing disease, as long as it stays put where it's supposed to be. It's usually harmless, even beneficial in their usual sites, because they will inhibit the growth of pathogens or participate in bodily functions such as digestion of food. Approximately 95% by weight of your feces is bacteria, but as long as they stay in your intestinal tract, they're good, and they break down the food so that these nutrients can become soluble and go to all parts of your body. Normal flora can cause infections if they are introduced to other sites of the body. For example, your skin is covered with normal bacteria, which prevents pathogens from becoming established in healthy individuals. So it protects your skin. If the skin bacteria enters the bloodstream of an immunocompromised individual, it can cause a severe, potentially life-threatening infection. As I said at the beginning, the result of any laboratory test is only as good as the quality of the sample provided. It is crucial that when a sample is collected, it is as free of contamination with normal flora as possible. It is not possible to detect contamination, so it's up to the individual in charge of sample collection to follow, or have the patient follow, the protocols exactly as they are written. Normal flora will cause issues if introduced into a culture sample, as the normal flora may overgrow and mask any pathogens that might be present. The most problematic sample is blood cultures. There's a specific cleansing process prior to collecting the sample that must be followed before the venipuncture is performed. And that's because the little plug of skin, when you insert the needle and then you start collecting the sample, there's a little plug of skin that will go into the container. And if you don't cleanse properly, then this will have skin contaminants that will easily overgrow whatever is there. In the old days, we would assume that skin bacteria growing in a culture were contaminants. Unfortunately, patients with compromised immune systems such as HIV may become infected with skin contaminants, and this would be a true pathogen in that particular situation. So you can't just assume. I had a nurse say, well, the lab can tell if it's a contaminant or not. No, not anymore, because normal flora can become a pathogen if it's introduced into the wrong parts of the body. Once a sample is submitted, it must be placed in an appropriate growth environment. The mantra for microbiology is out of the patient onto media, such as agar, as soon as possible to prevent the potential death of pathogens which are present. Usually we will get these samples on a swab and we put them in a little transport medium, but if we don't process it very quickly, these will die. Bacteria don't survive very well outside of the body. So we will plate it on agar, and the purpose of using agar is to provide an environment that's rich in nutrients, which will keep the organism, if present, alive. The sample is streaked onto an agar plate, and these contain the food for the pathogens we wish to isolate, and in some cases, inhibitors to prevent the growth of certain normal flora. There are many, many different types of agar plates, which contain a variety of additives. And the number of different types of agar media that you will use or streak the organism on will be dependent upon the source of the sample and the type of the organism, whether it's a bacteria, a fungus, or a yeast. Once the appropriate media is selected, the sample is spread or streaked onto the agar in a specific pattern. The goal is to have a section of the plate where there are single colonies which can then be tested for identification and antibiotic susceptibility. The plate is divided into quadrants, and the sample is applied then successively streaked across the different quadrants. So you notice that there's the thickest part, and this is where we started. And then we fire our loop to sterilize it, and then do another streaking pattern. Turn it, sterilize our loop, do another streaking pattern. And by the time you get to the fourth quadrant, hopefully you have streaked enough to where there will be single colonies, because you can't tell from this massive mess what the morphology or characteristics of this bacteria is. Gram stains are performed once the culture grows and individual colonies can be tested. And this is a method used to distinguish and classify bacterial species into two large groups, gram-positive and gram-negative. Gram-positive organisms retain the stain and appear violet or purple under the microscope. Gram-negative do not retain the stain and they appear pink. The colonies spread onto a microscope slide, stained, then observed under the microscope. In addition to the gram stain property, the organisms are also classified as to their appearance, and there are two basic forms of bacteria. Coccus, which the plural is cocci, whose overall shape is spherical or round, and bacillus, the plural is bacilli, is used to describe any rod-shaped organism, and this is like a boxy type of shape. And so you can look at the illustration and see that we do have our cocci and our bacilli, but there are other types of morphological characteristics that may be observed depending upon the type of bacteria we're looking at. The microbiology lab can be broken down into sub-departments. Bacteriology, which is the study of bacteria through culture, biochemical testing, and sensitivity testing. Sensitivity means sensitivity to drugs or antibiotics. Virology is the culture and study of viruses. Parasitology is the identification of parasites from a wide variety of samples, although feces is the primary sample that they're excreted in. And mycology, the study of fungi. Due to the complexity of testing involved and low volume, Most clinical laboratories will not perform virology, parasitology, or mycology on site. These are sent to reference labs. In order for you to stay up to date and current in your knowledge of identification, you would have to do these different tests every day. Because otherwise, if you only did them like once a week or once a month, if you don't use it, you lose it. Your knowledge will fade. Most bacteriology testing is performed in hospitals or reference labs. Hospital systems in many areas of the country are consolidating microbiology testing at a single hospital site so that it's more of a reference lab servicing many sites in the system. Testing is expensive and requires individuals with a high degree of competence. Practitioners require a very high level of critical thinking, application of knowledge, and decision-making skills when analyzing test results. Failure to identify a pathogen may result in serious medical consequences for the patient. And that's why I love micro, because there is a lot of thought being put into every plate that you read. The types of testing performed will be dependent upon the clinical facility. If a clinic decides to set up a small micro lab, they may do testing based on their specialty. If they are obstetrics and gynecology, the focus would be on sexually transmitted disease. If it's a general practice, throats and urines, because throat infections and urine infections are very common. Tests performed in-house must have a relatively high volume or it becomes a monetary liability. Lab testing is expensive. And so if you're not, if you only do one throat culture a week or one urine culture a week, you're not going to make enough money off that to make it worth your while. So culture is the top picture. And again, we discussed this as spreading the sample on appropriate agar and observing for growth. Certain microorganisms will have a specific smell aiding in their identification. For example, Pseudomonas, they say smells like grapes. I never got it. It didn't smell like grapes to me. You will learn to look at the size, shape, color, and consistency of the colonies on the different types of agar to assist in identification. And what's illustrated here is called blood agar, and this is made with sheep blood. They don't kill the sheep. They actually they do little donations, the sheep do, because they want them to keep on going and providing the blood. Then there's one called chocolate agar, and it does look like chocolate, but don't eat it. It's blood agar that has been cooked. And so when blood gets cooked, it turns brown. And then there's a lot of clear agars, and again, they add nutrients or inhibitors to them, determined by what we're looking for. The sensitivity is shown on the lower picture, and after isolating the pathogen, you make a solution, a specific solution of the organism, and pouring it onto an agar plate, which then you add antibiotic discs. And then you observe for no growth around the disc. And this indicates that the organism was killed by the antibiotic. The larger the zone around the antibiotic disc, the more effective the antibiotic. So if you look at the plate, you can see really large zones where this indicates the clarity of the zone, indicates that the bacteria has been successfully killed. If you look at this antibiotic disc, it's useless. It is absolutely useless. And even this one, it needs to be of a specific size, the zone, before it will be called out as the bacteria is sensitive to that antibiotic. These are reported out to the doctor so they can make a decision of what antibiotic is best for their patient. Virology, many viral infections such as flu, hepatitis, human immunodeficiency virus, can be diagnosed through traditional laboratory testing methods. Molecular testing is playing a larger role. In picture 1, the sample is collected. And again, there are specific collection kits just for viruses. The picture 2 is a viral culture. They're grown in cell types that the virus being tested is able to infect. Because remember, viruses cannot live by themselves. They must infect a host cell and be able to replicate, and then what they do is they explode, and then they will infect additional cells. And sometimes it takes about a month for a viral culture to grow. And the lower picture is the electron microscope, which is used to identify. And every virus has a different morphology. And the individuals who do electron microscopy, this is what they do day in, day out, 8 hours a day, 5 days a week, because there are so many viruses out there, and you really have to have the ability to look for specific small differences in order to classify a virus. Cytomegalovirus, varicella zoster, and rhinoviruses are some examples that may be grown in culture. Parasitology. Oh my goodness, this is one of my favorite courses to teach. Parasites are present in the United States and are transmitted through a variety of mechanisms: food, water, fecal contamination, and vectors such as ticks and fleas. Worms and amoeba are the most common, and due to worldwide travel, reference laboratories will see parasites not normally found in the U.S. And if you want a break from studying, please, please, please go to Monsters Inside Me. These are fascinating episodes about parasites people unwittingly become hosts to. And I used to watch this show all the time. So if we look to the right, there's the roundworm, and it's usually in the feces. And some of them are so large that they can actually be seen in the fecal specimen. And it scares the hell out of the patient because oh my God, there's worms growing inside me. Hookworm, look at the mouth on that guy. And they attach to the intestinal tract, and the teeth are very small, so they don't really hurt you, but they can cause severe damage. Then we have the fluke, we have tapeworms, and this tapeworm, they do grow long, and they have the very end, the scolex, is where they attach to the intestinal wall, and every now and then they will let go of proglottids, and these are pieces of the tapeworm, and you can actually see them, and they will actually move. Then nematodes is another type of worm, and then protozoa are very small. So again, parasitology, one of my favorites. If you work in a state health department, you will be doing parasitology. Mycology is the study of fungi, and they are beautiful. It's one of my favorite courses to teach, although we just touch superficially on the main types of fungi that cause diseases. Fungi can cause a variety of infections in various parts of the human body. In the mouth, it causes thrush, which the causative agent is Candida. And when my daughter was an infant, I thought she had milk stuck on the roof of her mouth, so I was trying to scoop it away, and it wouldn't scoop. She had thrush, and it's very common in infants. Athlete's foot, Tinea pedis, is the fungi that's a causative agent. Skin, you can get ringworm, and this is a fungal infection, and trichopiton is the organism responsible. And in the lungs, aspergillus. You can inhale these spores, and they can go into the lungs, and if you're immunocompromised, meaning your immune system is very weak, you can develop infections with this type of organism. Fungal infections of the skin are usually easily recognized, but sometimes cultures are needed, especially for internal infections. Cultures are plated on special media designed to grow the fungi, and they're observed for growth. Once grown, they are observed microscopically for identification. You look at them macroscopically on the plate, but the real identification is done at the microscopic level. Fungi are uniquely beautiful, and I encourage you to go to drfungus.com. It's a wonderful site, and they keep it updated, and it has beautiful pictures of just about every fungus that you can imagine that cause disease in man and woman. The world of microbiology is rapidly changing, and technology is playing a huge role in the testing processes performed in microbiology. Traditional culture and sensitivities take a minimum of 2 days to weeks or even months in the case of some fungi to get results. Mass spectrometry is rapidly becoming routine in large microlabs to identify organisms grown in culture. Molecular testing has become routine for many types of bacterial identifications, even in large physician office labs. I oversaw a lab that had a small machine that did a molecular test for gonorrhea and chlamydia. I want to spend a little bit of time discussing moldy top. Large micro labs are replacing biochemical methods of organism identification with matrix-assisted laser desorption ionization, or MALDI top for short. The principal microbes are identified using either intact cells or cell extracts, which are mixed in a matrix. So we grow it up, and then we take the colony and mix it in a matrix. The matrix is placed on a target plate and shot with a laser, which generates ions. Measures the time it takes for ions to reach the detector, and the data obtained is compared to a database to identify the organism. The advantage over traditional methods are that it is rapid, sensitive, and economical in terms of both labor and cost. So think about it. We have to do the culture, but the biochemical testing and other types of testing that are very time consuming and add to the cost don't need to be done because the machine does it by a different type of methodology. The limitation is that the fingerprint of the organism must be in the database, so there is a potential to miss new or emerging pathogens. So we always have backup systems. If MALDI doesn't give us an answer that is right, then we can go ahead and do the traditional testing. This picture is FYI in case you wanted more information. There are a variety of websites that can give you more in-depth information. You will learn the specifics of this relatively new technology during the microbiology course. And it's really only been out, I want to say, really gained ground in the last 5 years. So it's really new. In our world, that's really new. So if the very top left picture is getting the colony, putting on the matrix, then you put it in the machine, then it goes ahead and analyzes it and gives you a result. Very, very cool. Molecular testing in the clinical lab has exploded. Molecular testing has many applications: the diagnosis of genetic defects, determining the presence of infectious agents, and quantification of infectious agents such as HIV and hepatitis C. HIV, to make sure the drugs are working, we want to do a viral load and see that the viral load is decreasing. They now have a cure for hepatitis C, so molecular testing allows us to see that the virus is gone. The molecular testing can be performed directly from a clinical sample. There's no need to grow it up in culture. The genetic profile of many infectious agents has been identified, and testing is rapidly being developed for a larger number. The testing is becoming black box, where the samples are placed on an analyzer, which performs all the required steps. The primary downside of molecular testing is that most tests are still very expensive. And so by black box, when I've done molecular testing in student lab, I've taught the students how to do it, and it was very tedious, very time consuming, very labor intensive, and you had to do things just right. So you had to have your sample, you had to do PCR is still done. Polymerase chain reaction is like a Xerox machine for DNA, and it's amazing. And so we are able to take a sample with a minute amount of virus and put it through the process of the PCR, polymerase chain reaction, and end up with billions of copies. So this makes it easy then to perform testing and identify the particular virus. Now, those steps can all be done in a black box. The following is a list of frequently performed testing and the sample required, and there are many, many more. Sexually transmitted diseases such as HPV, Neisseria, herpes, chlamydia, and we can use urine, vaginal, or urethral swabs. Human immunodeficiency virus, that would be blood. Hepatitis C is blood. Methicillin-resistant Staph aureus is a nasal swab, and this is a very common test done in hospitals because you can be a carrier and not know it, and hospitalized patients are very susceptible to this infection. Enterococcus is a rectal swab, Clostridium difficile is stool, and group A Streptococcus is throat swab. The limitation of molecular testing, though, is that it cannot distinguish between living and dead organisms. If a physician wishes to determine the success of a treatment, a traditional culture will need to be done. In summary, in my opinion, microbiology is one of the most fascinating fields of study due to the variety of departments one may work with. It's becoming automated, but currently most laboratories continue to use traditional methods. It requires a very high level of critical thinking and decision-making in analyzing the results of testing performed. Unfortunately, the department stinks, but if you get bitten by the love for micro, you get used to the smells. And I really wish we could have done a lab tour because you would know what I'm talking about. When you're in the micro department because of the smell of all these bugs growing and the fumes that they create.